And welcome. We finally made it to my favorite unit of the year. We're looking at gene expression and regulation. So, big overarching idea for this lesson is that an individual's genotype is physically expressed through their phenotype. This topic one is going to be short. We only got a few slides here. Our objectives are to describe the structures involving and passing hereditary information from one generation to the next, and to also describe the characteristics of DNA that allow it to be used as hereditary material. So if we scroll down, you'll see it is a big unit, but we are going to be starting our notes here on page 8. So here we go. DNA and RNA structure. So we're going to dig deep, go back to unit 1. We're going to remind ourselves a little bit of DNA and RNA structure. Let's first talk about how it was discovered. So in the 1950s, there was a scientist, Rosalind Franklin. She performed what was called an X-ray crystallography of DNA. A lot of individuals like to give credit to Watson and Crick. All they figured out was the base pairing. The A goes with the T, the C goes with the G. There was this problem of the structure of it. If that were the case, then how do we get this double helix? So what she did was she did this X-ray crystallography on a piece of DNA, and it revealed a pattern that was repetitive and it was regular. So what she discovered was, I should say, as she was discovering that, Edwin Chargaff analyzed these DNA samples from different species. So at the time, we did not know if we had the same DNA as other species. He found that this rule holds true for all species, which is mind-blowing if you think about it. For you, for me, for your pets at home, the bananas, the grass, these rules hold true for everything. The amount of adenine is always going to equal the amount of thymine. They pair up together. Let that sink in. Adenine is always going to equal the amount of thymine. So if I tell you this piece of DNA is 25% adenine, you're going to say, oh, it's going to be 25% thymine. And the amount of cytosine is always going to equal the amount of guanine. So if I tell you there's 33% guanine in this sample, you're going to say, oh, I know how much cytosine there is. So nucleotide structure, this is unit 1 stuff. Dig deep, remember, unit 1. We have our purines up here. Our purines have a 2-ring structure. They're pure. They got the 2 rings, pure. These are your adenines and your guanines. So these are your A's and your G's. And then you have your pyrimidines. Your pyrimidines are a single ring structure. These are your CUTs. We cut pyrimidines. CUTs, we cut pyrimidines. So our pyrimidines are our cytosines, our uracils, and our thymines. You may notice here that as the nucleotides pair up, they pair up together by these hydrogen bonds that are in between each one. But I'd also like to point out that the purines match with the pyrimidines. Let's go back one. The purines match with the pyrimidines. So you're never going to have a 2-ring structure on each side. One side is going to have a purine, one side is going to have a pyrimidine. They do not bind together. The purines bind with pyrimidines, pyrimidines don't bind with pyrimidines, right? So if you remember one, you can remember the other. All right, but CUT, cytosine, uracil, thymine, those are your pyrimidines. Cool, check out this base pairing. They're held together by these hydrogen bonds. Boom, 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 boom. Remember, hydrogen bonds are weak when they are singular, but when they come together, they're strong in numbers. You'll also notice here that this adenine-thymine binding here, there's only 2 of them. Whereas in your guanine and your cytosine, they're bonded together by 3 hydrogen bonds. So because hydrogen bonds are strong in numbers, you'll see that the bonds that keep together guanine and cytosine are a lot stronger than the bonds that keep together adenine and thymine. This is going to be important later in this unit when we talk about DNA synthesis, replication, and DNA transcription translation. There you go. Adenine and thymine have 2 hydrogen bonds, and cytosine and guanine have 3. So it's a 2 to 3. All right, let's quickly review. Think ahead to DNA replication. How might hydrogen bonds aid in this process? Boom! Hydrogen bonds allow for the DNA strands to be easily separated during the replication process. So, piece of DNA, 2 strands, held together by hydrogen bonds. If we are unzipping the DNA one at a time, it's easy to break apart, but if we try to rip it all at once, it's going to be tough, right? So these are easily separated because of these hydrogen bonds. We just sort of unzip the whole thing. Quick review number 2. A DNA sample is analyzed and you find that 22% is cytosine. How much thymine would you expect to do in the sample? This is a trick question because cytosine does not pair with thymine. Here we go, 28%. Miss LaGraves, how the heck did you find that? Well, we have, let me just get my pointer. Here we go. We have 22% cytosine. Cytosine pairs with guanine. So that means that we have 22% cytosine, 22% guanine. If we add those together, we get how much? We get 44%. So 44% of this piece of DNA is this CG mix. But they are asking us how much thymine we expect to be in there. Well, how we do that is we take the 44% that's C and G, we subtract it from 100% of the DNA, and we come up with, that's right, 56%. We're going to divide that in 2 because, again, they're looking for the thymine, and thymine pairs with adenine. So we got 28%. Cool. So how did we discover the structure of DNA? Watson and Crick took the findings from Franklin. This is what drives me nuts about Watson and Crick. They took the information that Rosalind Franklin did to discover the 3D shape, and they took the work that Chargaff did for his base pairing, and they put it together and created the first 3D model. This is why it's important to put your name on your homework. Someone else will come along and take all the credit for it, and boom, now we have a double helix model of DNA. Okay, so it was really these 2 scientists working off of the work of these other 2 scientists, and that's how we discovered DNA structure. Some things to note. Number 1, DNA is double-stranded. We know this. This is Unit 1 stuff. 2 strands. The backbone is called a sugar phosphate. It's also a great name for a metal band with the sugar phosphate backbone. That's what we call it, the sugar phosphate backbone. It's that thing right over there. In the center of it, you have nucleotides that pair up. Adenine's pairing with thymine, guanine's pairing with cytosine. The DNA strands are anti-parallel. That means that one runs in one direction and the other runs in the other. DNA has directionality to it. We'll get into that. But there's a 3' direction here and a 5' direction here. So there's directionality to this. There has to be. How are we going to know which strand we're going to replicate which way? So one strand runs 5' to 3', that's this guy in the bottom over here, and the other one runs in the upside down opposite direction. So just know that they are anti-parallel to each other. In your 5' end, there's a phosphate group. Remember those phosphate groups that we see hanging off of things? That's in your 5' end, and your 3' end has a hydroxyl group. That's your OH. Oh yeah, you're not getting rid of Unit 1. We still need to do our functional groups. Right, so anti-parallel. Some key functions of DNA. These are things that we know. We've reviewed this before. This is going to be easy. We know that DNA is the primary source of heritable information. Genetic information is stored and passed from one generation to the next through DNA. If you're familiar at all with computer coding, the 1s and the 0s, the binary code, we can get a computer to do anything we want by using these 1s and 0s. Your DNA is the same, except we have those extra 2 pairs. Instead of 1s and 0s, we have A's and T's and C's and G's. Same kind of idea though. There is an exception. Because this is a biology class, we talk about all things living, not just humans. RNA is the primary source of information for some other prokaryotic cells, some viruses, some other like lower order organisms. So prokaryotic versus eukaryotic DNA, again, Unit 1, this is a review. Unit 2, this is a review. In eukaryotic cells, DNA is found in the nucleus, a membrane-bound nucleus. So there's a membrane around it. In order for us to get to this DNA, we must get through that nucleus first. They're also combined and condensed into chromosomes. So we have to relax and denature those chromosomes in order to get to that DNA to work with it in the first place. The eukaryotic cells also have linear chromosomes, so there's a direction to it, there's a directionality to it, but there's also a beginning and an end. So we can start on one end and we can go to the next. In prokaryotic cells, you guys know DNA is in the nucleoid region. There's no membrane bound to this. It's just kind of floating in the cytosol in the nucleoid region. And the chromosomes in this case are circular. This is beneficial to the prokaryotic cells because instead of having like a start and a stop, as in a linear chromosome, we can just plant some replication players right in the middle, and it can just go around and around and around and around. In prokaryotes and some eukaryotes, we have plasmids. We worked with plasmids when we were doing our E. coli lab, right? But plasmids are just these separate, non-nuclear, non-chromosomal DNA that can go through transcription translation to express various genes. There you go. There's the definition. Small circular DNA molecules that are separate from chromosomes. So let's talk a little bit about plasmids. Plasmids replicate independently from chromosomal DNA. So you can see the chromosomal DNA over here in the nucleoid region of our prokaryote, and then we have our plasmids over there. Plasmids are primarily found in prokaryotes. Some eukaryotes have them, but not too much, so we're going to disregard. It contains genes that may be useful to the prokaryote when it's a particular environment, but may not be required for survival. So this is your quorum sensing that we talked about. This is being able to regulate things in the environment or regulate their own protein expression through changes in the environment and be able to send out those signals really rapidly and quickly. They can be manipulated in laboratories. We did that. You and I, we did it. We took E. coli. We took a GFP protein that's sitting on a plasmid. We put it in and we made E. coli grow. What we do with this is we call this recombinant plasmid DNA. So just like we have recombinant traits in our non-Mendelian genetics, now we have recombinant plasmid DNA. So it can be removed from the bacteria. We can toss in our own genes that we want, recombine different genes, put it back into our bacteria, and voila, now we have E. coli that is producing a green fluorescent protein normally found in jellyfish. Pretty wild. But recombinant plasmid DNA is we are recombining genes. When the recombinant plasmid is inserted back into the bacteria, that gene will be expressed. Again, we saw that in our E. coli bacterial transformation lab. Very cool stuff. Bacteria can exchange genes found on plasmids with neighboring bacteria. That's our quorum sensing. Once DNA is exchanged, the bacteria can express the genes that are acquired. So it's like, hey, there's a change in the environment here. I have some genes that will allow us to give us more adaptation into this changing environment. Why don't you take it? This helps with survival. Big time. Here you go, in all of its glory. Main structural differences between RNA and DNA. Take it all in, pause the video if you have to. God love it. Here's some main differences. Number one, RNA is ribonucleic acid. It is single-stranded. And instead of thymine, we have uracil. So in RNA, A always goes with U, C always goes with G. If I present to you a strand of nucleotides and you see that there's a uracil in there, there's a U, and I ask, is this RNA or DNA? You would correctly say RNA, right? Because of the U. DNA, on the other hand, we call it deoxyribonucleic acid. Not a big deal. It is double-stranded, and we have that thymine in there. So if I gave you a sequence of nucleotides and it has thymine, and I asked you to identify whether it's RNA or DNA, you would correctly respond, that's correct, DNA. Let's do a quick review. What is Chargaff's rule? It was stolen by Watson and Crick, by the way. If you correctly said A pairs with T and C pairs with G, you're right. So what comprises the backbone of DNA? This is my metal band name. Sugar phosphate backbone. Yeah, it's your sugar phosphate groups on your DNA, the backbone of DNA. We'll see that when we start to build our own. Which nucleotides are purines? Which ones are purines? Remember, purines have two rings. That is your adenine and your guanine. Your purines are going to bind with your pyrimidines. Purines don't bind with purines. You gotta switch it up. What type of bonds hold together nucleotides? They're super strong when you have a whole bunch of them, but one by one, they're not very strong. That would be hydrogen bonds. You're right. Congrats. Couple more. Summarize the scientific contributions of Chargaff, Franklin, Watson, and Crick. You don't really have to know this, but it's always good to know as a scientist. Chargaff did the base pairing, Franklin figured out the helix shape, and Watson and Crick put it all together. Key differences between RNA and DNA given to me. Boom. DNA is double-stranded. Adenine bonds with thymine. RNA is single-stranded, and adenine binds with uracil. Those are the two key differences in their structure. Practice free response question. Did we give you the answer? No. So I'm going to read this out. You're going to do it as part of your notes. I can't wait to see your answer. Many species of bacteria are becoming resistant to antibiotics. Recently, studies have analyzed the chromosomal DNA of antibiotic-resistant bacteria. One bacteria studied is methicillin-resistant Staphylococcus aureus, also called MRSA, M-R-S-A. Researchers have found that its chromosomal DNA does not contain the genes for antibiotic resistance. They also found that when MRSA is cultured with non-resistant bacteria, the non-resistant bacteria become antibiotic resistant. For A, I need you to identify the location of the antibiotic resistant genes and discuss how the antibiotic resistant genes are being transmitted to the non-resistant bacteria when we culture them together. Congratulations! You just completed your notes. Thank you so much. I will see you in class.